Hello, I'm Julie Price, Professor of Radiology at Massachusetts General Hospital and Neuroimaging Researcher at the Martinos Center for Biomedical Imaging. I thank members of the American Chemical Society and symposium organizers for the honor of participating in this special symposium, recognizing the pioneering work of Dr. Saul Hertz and designation of MGH as a National Historic Chemical Landmark. Dr. Hertz's seminal advances in the medical use of radioactive iodine 80 years ago, coupling in vivo imaging with therapeutic dose delivery, still drives the innovative spirit in nuclear medicine and molecular imaging today. I will speak about current uses of radiopharmaceuticals in brain imaging and review key advances to highlight the importance of neuroimaging in nuclear medicine and healthcare. The neuroimaging modalities I'll discuss today, described on the left, are based on emission tomography, positron emission tomography or PET, and single photon emission tomography or SPECT, provide us with functional measures, and magnetic resonance imaging or MRI provide us with structural or anatomic information. On the right are examples of functional neuroimaging outcomes and example clinical applications. The outcomes range from general physiologic parameters such as blood flow and glucose metabolism, or aspects of cellular function such as proliferation, or the study of enzyme activity that may be important for cellular signaling or regulation of gene transcription. These types of measures are important in many clinical applications, including neuro-oncology. We can also target specific functional proteins because of the research development of radiopharmaceuticals that bind with high specificity and selectivity to protein targets of interest. Examples in this domain might include targeting of synaptic function via imaging of the synaptic vesicle 2A protein, or imaging of translocator proteins to study the brain's response to inflammation. We can also image neurotransmitter system functions such as serotonin, dopamine, and opiate. Shown in this category is an example of a PET image obtained using a radioligand that binds specifically to dopamine receptors that are rich in striatum, the dopamine system being of great importance in addiction and Parkinson's disease also, where there's progressive degeneration of the dopaminergic system. The last example is in vivo imaging of misfolded proteins relevant for Alzheimer's disease, namely amyloid beta plaques and neurofibrillary tangles containing tau. The image in this category is an example of widespread amyloid plaque deposition in an individual with Alzheimer's disease. After sufficient validation, if neuroimaging can detect a biologic feature that provides information relevant to diagnosis or another aspect of the disease process, it can serve as an imaging biomarker. There is much that imaging biomarkers can tell us relevant to healthcare, as nicely summarized by Dr. Saranen and colleagues. There are several classes of biomarkers. We're familiar with the diagnostic biomarker that tells us if a disease is present, but phenotypic biomarkers tell us more about the biological behavior, with prognostic biomarkers helping us to answer questions related to outcome and disease trajectory, with other examples relevant to therapeutic intervention and drug performance. As you know, some neuroimaging biomarkers or radiopharmaceuticals are approved by the U.S. Food and Drug Administration for specific use. To put things in perspective, on the left, FDG gained approval for oncology and epilepsy about 25 years after the approval of radioactive iodine therapy. On the right, several SPECT perfusion and blood flow agents are approved with stroke as a common use. Over the past decade, there have been multiple approvals of radiopharmaceuticals to target specific biological proteins affected by or involved in neurodegenerative processes. The SPECT DAT scan for striatal dopamine transporter visualization relevant for Parkinson's syndromes, and within the green box, 3 amyloid PET imaging agents and 1 tau agent that are approved for the evaluation of Alzheimer's disease. In the final slides, I will focus on this area and the evolving role of neuroimaging in Alzheimer's disease that has further established its importance in healthcare. In 2004, the Alzheimer's Disease Neuroimaging Initiative, or ADNI, was launched and is a landmark public-private partnership. ADNI has accumulated at least 15 years of clinical neuroimaging, cognitive biofluid, biomarker and genetic data, biofluid samples. These data are available to researchers, resulting in more than 3,500 publications. Key accomplishments of ADNI over the from 2000 to 2014 are listed here, and I'll highlight some that I think are very important in healthcare. This includes standardized biomarkers for clinical trial subject selection, standardized protocols for multi-site studies. It's also inspired initiative in brain injury and post-traumatic stress syndrome and depression as AD risk factors. It served as a data sharing model, which is very important in terms of information dissemination. And it's inspired other partnerships in other neurodegenerative disorders. But the vast amount of information has led to the identification of novel AD risk alleles and understanding of relationships between biomarkers and AD progression. This supported the development of a hypothetical model of dynamic biomarkers of the Alzheimer's pathological cascade. The model stressed the importance of focusing more on biological markers in early disease detection where therapeutic benefit may be greatest before significant neurodegeneration. In the cascade on the left, the amyloid deposition occurs first, and that's followed by synaptic dysfunction and tau-mediated neuronal injury indicated by the orange and green curves. This is followed by structural brain changes shown in blue. And all the way to the right, cognition and clinical changes are the last to be detected in the cascade. This body of work led to a joint National Institute of Aging Alzheimer's Association task force aimed at revision of diagnostic and research criteria for AD, including a preclinical stage. In 2018, there was a framework published envisioning a definition of AD as a biological construct. To provide further context, this slide summarizes information offered by amyloid and tau PET across the Alzheimer's disease spectrum, from cognitively unimpaired to those with mild cognitive impairment, or MCI, and AD. On the top, you see amyloid appearing early in frontal cortex and in subcortical areas with very advanced disease. Some who are cognitively impaired accumulate amyloid with heterogeneous uptake in MCI subjects, some of whom will develop AD, and some can bear high AD levels of amyloid without carrying a dementia diagnosis. On the bottom is tau PET images that are consistent with neuropathological Brock staging across the AD spectrum, with early levels of tau detected in inferior temporal lobe on the left and eventual spread to isocortex in advanced disease. The role of amyloid and tau PET in clinical trials continues to evolve in terms of eligibility criteria and endpoints. There's been great effort in international public, private, collaborative, and industry partnerships targeting prevention, daily practice, and drug development in AD. Examples of earlier clinical trials utilizing amyloid PET include the large longitudinal multi-site IDEA study of Medicare beneficiaries with MCI or dementia of unknown etiology. Rabinovich and colleagues reported that the use of amyloid PET in these individuals was associated with changes in clinical management. And there's several European initiatives launched in prevention trials that focus on preclinical or inherited Alzheimer's disease. Drug development efforts are now largely focused on disease-modifying therapies rather than symptom management. This summer, the FDA approved an anti-amyloid agent, aducanumab, that selectively targets and removes amyloid beta plaques, ushering in a new era for AD therapeutics. In closing, it is expected that there will be a general paradigm shift in how neurodegenerative disease are diagnosed and treated, with a future role of PET imaging in this context. Rather than syndromal context, there'll be biological constructs. It's expected that new radiopharmaceuticals will emerge to effectively target neuropathology in other diseases, such as alpha-synuclein in Parkinson's disease and dementia with Lewy bodies, and TDP43 protein in ALS and frontotemporal dementia. I will end by remembering that advances reviewed today are accomplishments that were made while standing on the shoulders of giants, those who made such work possible and meaningful. It is an honor to stand on the shoulders of Dr. Hertz. Thank you.